Hi guys, good morning. I am Dr. D.R.T.SUMATHI, Assistant Professor, Department of Zoology. Today I am going to take Developmental Biology, the subject, first unit, Module 1. Just you have to know about first unit. Next. Normally, Developmental Biology. What is meant by Developmental Biology, and also why we have to know about Developmental Biology? In my opinion, we should know about developmental biology. That's very important and also very interesting subject, because just you have to know about myself and all living organism. Normally developmental biology is otherwise called embryology, and also it is one of the field biology, and it developed from unicellular organism, and also it goes to multicellular organism. Just you have to know about all the growing. Which one is controlling the growing? The genes. The genes are also very important for growing. Just we have to know about nucleus, genes and DNA. How to helping to developing the normal organism growing. That is a multicellular organism. Normally knowledge of the normal developmental process can aid to understanding the developmental abnormalities. In case we have the abnormalities, that condition is known as cancer. Normal cell divisions, you know, the mitosis, meiosis and also everything, but then the cell division is not occurs. The cell division in case multiplies, we cannot controlling the cells, that position is called cancer cells. Now Hans Sperm, that is one of the scientists, he got a Nobel Prize in 1935. That is a remarkable biologist in the 20th century, the modern experimental embryology. That is a founder of embryology. Next, just we have to know about some important terms in the developmental biology. Otherwise, we cannot understanding the technical term. What is meant by gamete? The gamete is called egg or sperm. It is indicating both egg and sperm. That is called gamete. Another one is a gametogenesis. Production of egg or sperm, that is called gametogenesis. Another one is a oogenesis. Production of eggs is called oogenesis. Another one, spermatogenesis. That is called differentiation of the sperm morphology, external morphology of the sperm. That is called spermatogenesis. Another one, follicles. Follicles where the egg is mature in the ovary, that is called follicles. Normally the egg is developing from our ovary. We have the ovary, pair of ovary. In monthly eggs, one pair of ovary also producing one type of egg, that is called OVUM. Ovulations, that is releasing of the egg from the follicle cells, that is called ovulations. Another one is a polar body. This polar body is a non-functional product during the meiotic division in oogenesis, that is called polar bodies. Only oogenesis we obtained the polar body. In spermatogenesis we didn't get the polar bodies. Another one is a zygote. The zygote is otherwise called fertilized egg. When the sperm is fused, that is called fertilized egg. Next, this is a structure of human sperm. What is the structures? The normally in the external structure, head region, body region and also tail region. All the organism also have the head region, body region and tail region. In the sperm, human sperm also head region. The head regions contain some crescent shaped, that is called agrosomes. In remaining slide, I will explain what is meant by agrosome and the role of agrosome during the spermatogenesis and spermiogenesis also. Another one is the important nucleus, because you know very well about the nucleus. During going to the cell division, there are so many stages, mitotic, meiotic stages also occur, the DNA also occur, all the content also occurs in the nucleus, and the nucleolus also present in the nucleus. Our body is made up of cells, each and every cells have the nucleus. And this nucleus and agrosome, both of them is called head region. Another one is a neck regions. That neck region is the middle piece. That middle piece also contain a mitochondria. Mitochondria, which is shape is a spiral shape of mitochondria. The role of what is the role of mitochondria in human cells also, that is a electron transport system, another one is a metabolic activities, everything also important of the mitochondrion. and also it is covered by the plasma membrane. And this structures is called tail region. Third region is a tail region, that is it is fully covered by the plasma membrane. Next, this human ovum, the head, the sperm also contains a DNA material, and it combined with the egg's DNA will create a new individual. Normally it do not any function. It goes to touch with egg after they have to producing with DNA. It combined with this DNA creating the new individual, the starting from the new individual from the head region. That's very important. Another one is the tip of the sperm head is the portion is called agrosomes. That is, agrosomes is one of the important word in the sperm because which enable the sperm to penetrate the egg. These agrosomes also contain one of the enzyme. That enzyme agronuclei, that is uses to the enzymes is agrosomes enzyme also used to penetrating the egg. That is normally it is a piercing the egg. Another one is the mate bees. It contains a mitochondria, which supplies the energy to tail to need to move. It goes to move to egg. It's very, very important. So the mitochondria role is supplying of the energy during movement of the sperm. Next, this is a spermatogenesis. Just we have to know about sperm, how the sperm is productions occurs in our body. That it is very important. That is a 2N. In the picture it shows spermatogonia. That is a 2N. 2N means diploid nucleus, not haploid nucleus. After the diploid nucleus, it goes to mitotic cell divisions. Normally, mitotic cell division is a cells also occurs in a remaining. Meiotic division is a reduction divisions. During mitotic division, primary spermatocyte is formed, the first meiosis stage. After it goes to haploid number, that is called secondary spermatocyst. After meiosis occurs, not mitosis, just you have to remind meiosis. During meiosis, the cell division is stopped, that is spermatid is formed. You have to note it during formation of spermatid, only the one nucleus is present. So just we have to note it N, not 2N, 1. After 2 nucleus is formed in the one spermatid. Next one, spermatogenesis. And after spermatogenesis, the spermatozoa formed from the each spermatid. First this picture shows 2N, that is called a spermatogonia. That one spermatogonia is produced the 4 spermatozoa, single sperm. Next, what is the process occurs above the picture? The spermatogenesis process. This spermatogenesis is the process where the sperm mother cells is seminiferous tubules. After testis, it is changed into the haploid spermatozoons. Already I explained that positions of the picture. Spermatogenesis is a formation of haploid spermatozoon. It is a continuous process. It is starting from the male to attain puberty stage and remain it continue till death. Normally it is producing the male. The process of spermatogenesis take place in the 2 stages. One is a formation of spermatid, another one is a formation of spermatozoa. What is meant by formation of spermatid? Just you have to know it. The spermatid is a haploid male gamete that result from the division of secondary spermatocyte, not primary spermatocyte. During primary spermatocyte, normal 2N is also occurs. During secondary spermatocyte, the spermatid is the haploid male. What is meant by formation of spermatozoa? Spermatozoa is otherwise called sperm, nothing but a sperm. The sperm are the male sex cell that carry the man genetic material. It is transferred from the man to the egg, that is called spermatozoa, that is called sperm. Next, spermatogenesis process. The formation of spermatid take place in 3 stages. 3 stages is related to division of the nucleus, that is a multiplication phase, another one is a growth phase, another one is a maturation phase. During multiplication phase, the cells also multiply. During growth phase, the cells also growing itself. Another one is a maturation stage, the cells also, new cells also formation. That is called maturation phase. During multiplication phase, The undifferentiated germ cell present in the seminiferous tubule of the testis, the spermatogonia or sperm mother cells also multiply. One and mitotic divisions also occurs. The germinal epithelium lining of the occurs in the seminiferous tubules. That times the spermatogonia have the prominent nuclei that contain 2n, that is a diploid nucleus. That diploid number of which one is a number, that is a chromosomes. Normally chromosomes is present in the DNA. The DNA is a genetic material. It transfers all the characters from one generation to next generation. But how many chromosomes occurs in our body? Totally 46 chromosome, 23 pair of chromosome, 22 pair of body chromosome and 1 pair of sex chromosome. That spermatogonia also contain a diploid number. That chromosome is a 46 in the human chromosomes. Next. Growth phase. This spermatogonia stop division during growth phase. Which one is a stop? Increases the size only. Size only increases by the accumulation of the cytoplasm in the nutritive materials. It became a primary spermatocyte, that is a 2n stage. The process is called spermatocytogenesis. Normally cytology means study about the plasma membrane and plasma, that is called spermatocytogenesis. The primary spermatocyte are double the size of spermatogonia. During primary spermatocyte, it is increasing the size, that is a spermatogonia also developed during the mitotic divisions. Next. Maturation phase. Third stage also matured. Matured means that all the cells are matured in stage. The primary spermatocytes to ready to meiotic division, that is called maturation stage. During mitotic division also occurs multiplication and growth phase. During maturation phase, the spermatocyte ready for mitotic division, not meiotic division, not mitotic divisions. The homologous chromosome start pairing, that is called synapsis. It is a crossing over. Each homologous chromosome split longitudinally by the chiasma formation. During formation of chiasma, they have to exchanging the genetic material. That is a genetic material exchanging is otherwise called crossing over. What is the mean by crossing over? During crossing over, changing the chromatid of the homologous chromosome. Normally A and B, A changing to B, B also interchanging of the genetic material from these ones, that is called crossing over. Another one is the DNA amount is duplicated, the time also beginning of the division. The time only the DNA also duplicated means that is divisions. At the result, what is the result? During the divisions, two cells is formed 4 spermatid. That is a maturation phase. All are haploid. That's very, very important. All are haploid in nature, not diploid. That is a haploid is indicating N, single N only, not 2N. Next. What is the significance of the spermatogenesis? What is the importance of the spermatogenesis? Each spermatocyte undergoes 2 maturation divisions. One is meiosis, first and second, and also it is produced 4 spermatid, the half number of the chromosome. It is necessary to maintain the number of the chromosome contained from generation to generations. Since normally the somatic cells contain a diploid number of chromosome, the sperm and the ovum also had the diploid chromosome. During meiosis, first the crossing over takes place. The formation of crossing over, they have to interchanging of the genetic material. It also bringing the variation through the evolutions also. That's very important. Next. It is entirely different from the human egg. Before slide, just we have to know about the human sperm. These slides also we have to know about human egg. Human sperm also present in the male. Human egg also present in the only female. Female have the egg. The human egg is otherwise called ovum. This is a structure of ovum. The egg also external structure, normally outer layer. The outer layer is also contain a corona radiata. All that is called the follicle cells. All the eggs also coming from the follicle cells. Next, the eggs also covered by the cytoplasm. That is a granules, that is called cytoplasm. All the cells also normally contain the center part, that is called nucleus. During formation, human egg that contain a first polar body. The first polar body, next it also contain a round circle, the blue color circle also, external circle, that is called zona pellucida. Zona pellucida is otherwise called jelly coat. One of the jelly coats surrounding the egg, that is called zona pellucida. But totally, and it also, human egg also contain the cytoplasm, corona radiata. That corona radiata is otherwise called follicular cells, and the zona pellucida. The zona pellucida is nothing but a jelly coat, that is a covering. Another one is a first polar body, next one is a nucleus. Next, that is how the egg is coming from that during ovulation. What is the production occurs during oogenesis? That picture shows a corpus luteum. Another one is a primary oocyte, primary follicle cells, that is a structure of ovary, and mature follicles, everything also occur. The ovary also producing egg. In case the sperm is not fused with the egg, and the ovulations also occurs. The after formation of ovulation, the menstrual cycles also take place. But what is the process occurs in the ovulation? The egg also coming from, egg also are ruptured and coming from that places. That is called ovulations. In the ovulation is not occurs, normally egg also divided from where it is. That is a primordial germ cells, and that diagram also explaining oogonium. That oogoniums also contain a diploid number. It is a 2n number. After that it also developed primary oocyte. Next one of the resting stage. Normally they have not preparing any one of the stage. That is called resting stage. That stage also called first meiotic division. After the formation of first meiotic division, the secondary oocyte also formed. The secondary oocyte after the preparation of the first polar body. Second meiotic divisions also occur in the secondary oocyte. The formation of the second polar body. During second polar body, only one egg is ruptured. The remaining 3 egg is not ruptured. That is it is a formation of oocyte. That is a haploid number. That is a ovum also haploid. In this diagram, we did it indicating the 3 polar body. After secondary oocyte, 3 polar bodies also formed. Only one egg is ripe. That ovum is a haploid. That is a fusion of the sperm. The remaining 3 also polar body. The polar body also nothing doing anything. That is called the polar body. Next, what is the stages occurs in the oogenesis? Already we know about the spermatogenesis. That is 3 stages also occurs. The same 3 stages also occurs in the oogenesis. First one is a multiplication phase. Next one is a growth phase. Next one is a maturation phase. And some of the term also varied, and some of the process also slightly varied. In this multiplication phase in the oogenesis, the haploid and the diploid germ cells are divided by mitosis to form the diploid oogonia in oogenesis, the spermatogonia in the spermatogenesis, oogonia in the oogenesis. Spermatogenesis is a formation of a sperm, but multiplication phase oogonia is a formation of oogonia. During growth phase, what are the stages occurs mean? This phase involves only growth in size of ovum. Next one, maturation phase. This phase involves meiotic division, and it divided into 2 phases, that maturation phases also. Next, oogenesis normally process formation of female gamete. It's very important, just you have to note it. The process is begin inside the fetus before birth. In mother womb also prepared for fetus, the process also beginning. That is amazing. It is starting from the mother womb, mother uterus also. The steps in oogenesis up to the production of primary oocyte, it occurs in the before birth, but the spermatogenesis is not occurring in the before birth. Only after birth, also after maturity stage also spermatogenesis occurs, but oogenesis it's a grade, that is a oogenesis starting from the mother uterus also. The primary oocyte do not divide further, and they either become secondary oocyte or degenerate, that time also. During oogenesis, the outermost layers of the ovaries also occurs, that is starting from the germ cells, that is called oogonium. Normally the oogonium undergoes a mitosis division. The mitosis is a increases in number of the cells, and the process of the oogenesis is take place 3 stages. Just we have to divide it. One is a prenatal stage, another one is a antral stage, another one is a pre-ovulatory stage. Just we have to know about what are the stages occurs in this one. This is a ovary or ovary structures and oogonium structures. For how the oogonia is developed during after ovulations. Normal growing of the follicle. First one is a primordial germ cell. Second one is a primary follicle cells. Third one is a growing of the follicle cell. Fourth one is a follicle cells divisions. Fifth one is a rupture of the follicle cells. Another one is a ovulation occur. That is a corpus luteum also participated, and normal disintegration of the corpus luteum, automatically the ovulation occurs. In case it is not occur, normally primordial germ cells, already I told you, oogonium developed from the primary oocyte. That time also 2n stages diploid in number. After secondary oocyte is participated, the first polar body also formed, and the next polar body also formed. The oocyte also formation of lastly. The ovulation does not occurs normally. The automatically in the egg also production also occurs in the ovary. That is what we have pair of ovary, but monthly cycles only one ovary is producing one type of egg. Next, that is a mitotic events occurs in the before birth and the childhood stage, after the development of the stages. The picture is very well shows about mitotic division and also growth phase. And the primary oocyte, the follicle cells involved oocyte formations, and the primordial follicle cells also formation during childhood stage, each month from the puberty and the menopause stage. After the development of the egg, the spindle formation occurs. After meiosis stage, primary oocyte developed from the each month, and the first polar body also formed. Meiosis, the polar body may or may not occurs. The second polar body also developed in the ovum, that is developed in the oocyte. During this picture shows the ovum is attached to the meiosis, the sperm penetration occur. It is developed from the one of the zygote is formed. That is a ovulations does not occur. Normally ovum is developed, ovum is fused with the sperm, it produces fertilization and the formation of zygote. Next, ovulations. Just we have known about the what is meant by ovulation. Already I told you, ovary is developing the egg. The egg is ruptured from the ovary, that is called ovulation. The development of the oocyte take place in ovaries. Every oocyte is neighbored by follicle cells from the follicle. As the menstrual cycle is start, the primary oocyte is initiated to grow bigger, and follicle cells also rise in number, causing the follicle cell grow larger to division of the larger cells. Normally some nutrition, so oocyte degenerate and leave just the follicles to mature and deviate from the cell. Fraternal twins may be born, which the distinct genetically. That is one of the different part of the formation of twins. Just you have to know about. Sometime it may be chances to born a twin. Some it is participating of the follicle cells. Some of the genetical variations. Next. During follicle cells attain maturity, the primary oocyte finishes, and the primary meiotic divisions also become. That is a developing of the secondary oocyte. Immediately the follicle breaks and the secondary oocyte is liberated from the fallopian tube, even when the second meiotic division has not happened. You know the fallopian tube, what is meant by fallopian tube? The ovary also have the two tube, that is called fallopian tube. After the formation of zygote, it comes to the fallopian tube. It is joining to the uterus. After formation of zygote in the ovary, it goes to fallopian tube and to uterus. That is called fallopian tube. That's very important knowing about the fallopian tube. This release of the secondary oocyte from the ovaries is known as ovulations. Releasing of the oocyte. Next. What are the process formation of female gamete? Normally oogenesis is the type of gametogenesis through which ova is also the female gamete are formed in the produced the female gamete is known as ovum. Ovum, female gamete, that is egg, all the terms also indicating the only one terms. In general terms, the female gamete are referred to the eggs, but the word egg can involve the various stages of development. Therefore, the significance of the egg is varied based on type of formation of organism. So, just we have to indicating the female gamete and egg and ovum. Next. For example, just we have to explaining what are the difference between the egg stages and ovum. Why we are differentiating that word. Once after the bird lays the egg, the entire development of the egg until the transformation of the egg into the chick occurs within the egg. Normally you know very well about what are the stages occurs in the egg embryo. The yolk materials also participated to formation of the egg materials, where the yolk materials content, that is a microlystal, macrolystal, mesolystal, that is a participation content of the yolk material, uses of the development of the chick embryo. Whereas the placental mammals, once the egg is fully developed in the fertilized, it began to divide and we no more call, and it is the egg. Just we need to recall that every ovum has to be haploid and consists of single copy of every chromosome. It is very important. Next one. The process of 3 stages, the prenatal stages and the antral stages, already I told you, just we have to know about. And the primary oocyte grows while being arrested the meiosis. During the cells also secreted one of the protein, that's very important, and it is called glycoprotein. Where it is present in the glycoprotein, it is a form of zona pellucida around the primary oocyte. Already in that diagram also showing the zona pellucida. During antral stages, the fluid filled with the flea area, the time present between the glucose cells and combined with some central fluid. It also contains some of the space, that is called antrum. So that is called antral stages. These are known as secondary follicles. In every month cycle, the secondary follicles is developed under the influence of the follicle stimulating hormone and luteinizing hormone. This hormone is very important for ovulations. FSH hormone, shortly we told the FSH hormone and also LH hormone, that is a follicle stimulating hormones. Another one is a luteinizing hormone. These both hormones also called sex hormones. It's very important during the ovulation and oogenesis also. Next. And pre-ovulative stage, that we have to know about LH, that is a luteinizing surge, meiosis complete here. That time also 2 haploid cells is nucleus and formed the follicle. One daughter of the cells receives less cytoplasm from the polar body. The cell does not participate in the ovum formation. Already I told you the role of polar bodies. The other daughter cells is known as secondary oocyte. The 2 daughter cells undergoes a meiosis. The polar body is replicated to form the 2 polar bodies, while secondary oocyte arrested the metaphase stage of the meiosis second. That time also formation of 3 polar bodies and 1 oocyte. That 1 oocyte is a ruptured egg. Next. Meiosis second is completed on the fertilizations. It gives off the 3rd polar body. If the fertilization does not occur, the oocyte degenerated 24 hours after ovulation while remaining arrested in the meiosis second, the cell divisions. In case the fertilization of the human sperm does not fuse with the egg, the fertilization does not take place. That time ovulation automatically occurs. Next. And that is the difference. Just we have to know about what is meant by spermatogenesis. Another one is oogenesis. The picture shows you very clear. The major difference between the oogenesis and spermatogenesis is there. Oogenesis is began in the fetus period to birth. It is a for oogenesis is formation. So oogenesis is starting from the mother uterus. That is very important. That is what is the difference between spermatogenesis and oogenesis? Normally primary spermatocyte, that is also primary oocyte. During meiotic divisions also occurs, secondary spermatocyte is formed. At the same time, the first secondary oocyte also formed. During secondary oocyte formation, polar body also formed. The first one is a first polar body also formed, another one is a second polar body is formed. Now that only one oocyte is formed and ovum also developed from the primary oocyte. But spermatogenesis, the meiotic divisions occurs, second meiotic divisions occurs, 4 spermatid is formed. That is 4 spermatid is differentiations and formation of 4 spermatozoa, and the 4 spermatozoa is otherwise called sperm, and each and every spermatozoa containing a single nucleus, that is a N. That's very important. Next, and that is a conclusions. Why we study about this spermatogenesis and oogenesis. Both spermatogenesis, formation of a sperm, and egg, that is called gametogenesis. This topic also gametogenesis. That gametogenesis is a significant part of mammal reproduction, through which the germ cells lineage undergoes a series of complex developmental process and forms fully matured gamete, that is a spermatozoa and oocyte. And that time also a number of conditions including infertility result from the normal circumstances of the gametogenesis. Without gametogenesis, any one of the organism does not develop. So it's very important to know about this gametogenesis. Thank you.